Okay. And we're recording. So, Doctor, what attracted you to your academic field, and what is your academic field? Because I'm meeting you today.
So myself, Dr. Mousami Majumder. I'm a professor at the Department of Biology at BU. So I teach biology courses. I teach both first year courses and also upper year courses, and also master's courses. So my specialty is in the research is breast cancer. So I'm a breast cancer researcher now, but I had done my PhD on oral cancer. So your first question is that what attracted me to the academic field? I would say that my curiosity, which was being nurtured and nourished by my professors. During my undergrad time, I was doing a small presentation. I was an honors student in microbiology at University of Calcutta, which is in which is in eastern part of India. And that university, we are doing our honors project. We're working with bacterial resistance, like how we develop those antibiotic resistance. And the way I was doing my research and presentation, my professor, fourth year professor, she told me, I do not know why, she told me that I strongly think that you are going to be a scientist in the future. Um, and I didn't know why she said that, but she could say that I'm extremely curious. And I kept asking questions, not just for my own research, but anything asking question is something I think fundamental. And your professor know actually you better by your seeing you for a few years, they know your interest, and that reflects on your writing assignments, paper, and etc. So during my undergraduate time, my professors at University of Calcutta, they actually motivated me to seek more and go for master's degree right after my undergrad. So then during my master's, I was involved in cervical cancer project at the biochemistry department at Indian Statistical Institute in Calcutta. So first time I had something interesting like cancer, and I was working on cervical cancer. As a microbiology student, my interest was virus, which is so popular now. And then I had a chance to screen those HPV. Now we have the vaccine, but my project was screening those as a master student in actual cervical cancer specimen. Again, my interests were more because I was open to an ocean of knowledge, and I knew nothing from something so open in front of me. And again, that curiosity kept driving me. So my professors are those who attracted me, and that my curiosity attracted me towards academic field. That's what I think.
That is very interesting that you came to Canada after going to school. Wow, you've traveled quite a ways. And so I guess that takes us to who influenced you most in your academic field. Was it a professor?
Yes, my professors. Again, when you are young, you are thinking of getting a degree, getting a good job. So being really a studious student, I was always on the scholarship at the university. All the time, all scholarships was there for me. And also at a time, my parents wanted me to go for higher education more, because I decided at some point during my master's that I am not going for medicine, but I'm going for more research, which is grad school. Uh, in India, systems are a little different. You do masters first, then you go for a PhD. So, but as microbiology degree with good marks, top student in the class, I got great interview from 3 companies as a microbiologist, and one actually offered me really good job right away on campus, that we are going to hire you in a big food industry. And before I finished my MSc, I had an offer letter on my table. So and right after my masters, I joined that company. But within 1 month, I realized that something is not fine with me. I got the good job. I have my master's degree. Uh, I'm settled, and that's a good thing to have a job and your education uh, back home in India, where higher education are always prioritized. And as a woman, I'm getting independence, and my parents are happy too. However, what do I do with my curiosity? So again, gone back on a weekend to Indian Statistical Institute with some of my friend who happens to be there at the spot over a weekend. They are being interviewed for PhD position, and I was there just to hang out and spend time with them. I was not interested in that program because I have a job. But that professor with whom I did my uh, MSc project saw me outside and asked me to go for this interview, that he thinks that I could do answer all those. There's a spot or walking interview going on. And again, for PhD, you get good institute. If you're accepted for a PhD, you're fully scholarship, and those scholarships are really reasonable for a student to have. So I told him that no, I'm not interested because I got the job. So but professor, just let me go and see the process. I went for the interview. I came home. I spent my weekend, and by Monday, I went to my job. I didn't have cell phone then. My mom called me. I came back from work. My mom called me that your professor called you from Indian Statistical Institute, and that Dr. Roy, who will be future my PhD supervisor. So I wanted to, as a courtesy, I went to talk to him, and he said that, well, you were selected for the PhD. I said, but I'm doing the job. He said, yeah, in the job, your salary would be 5 times or 10 times when you become, an adult, you reach that age. Here in academics, you might not have that much of salary right away, but it's an open end. Your curiosity can go sky high. There is no limit to that. And by that time, I was already bored, or I shouldn't say, I do not like that routine job at my workplace. And I was reading scientific article on more on cancer when I am 9 to 5 holding a job. So that line, I would say, that changed my entire life. Dr. Roy, when from Indian Statistical Institute, he said that here your salary will reach here. Here your curiosity is never going to be saturated, and at time you will be earning more than anyone else. And actually, 5 people like that will be working for you if you go for PhD and further education, and if you're a successful scientist. So I think that changed, and he's the one who take me towards the academic field. And I quit my job after 1 month, and I joined this position at Indian Statistical Institute. That time it was human genetics unit. Yeah.
That's really neat. That's nice that the prof like looked more for like your future rather like that was really nice of them to say that. That's really neat.
Yes, I think so, because you need someone to tell you or show you the path, and you need that mentor at some point. Beyond your professor, they know you, or they can see that spark in you, or know your motivation, or know that as a person how different you are. And it cannot be judged by GPA all the time. And that's what I think. Your interest, your curiosity is different than your academic degree and GPA.
Absolutely, yeah. So then I guess that answers the next question when you decided to pursue your career. So what would you say is the most enjoyable, exciting, or like the best part of your field of study? Like what is the most rewarding thing about it?
Not knowing the answer.
Pardon me?
I'm doing not doing the answer, meaning that I'm a scientist. I'm doing experiment to get some answer. So all the time I'm seeking for some answer. So in breast cancer research, what I do, we are trying to find out a marker which can detect cancer early. How early? Well, if it is detectable in the blood, then anytime you go for a physical checkup, we can ask young adults, not just 50 years old, to have a test for that. So if we can detect that earlier, we can actually have more time to treat the disease. So why I say that what is most exciting, that you are always finding an answer, you are trying to answer why this happened, how to resolve it, or is there a solution coming out of it? Meaning that if you know it already, you don't do the research, do you? In my case, I really like the way things proceed, and you try to find a solution or an answer on the outcome of your experiment. So that's very exciting for me. And knowledge, as I said, every day thousands article published, and there is no limit how much you can know. But at the same time, in your own field, my field is breast cancer research, as I said. Um, Again, most exciting part is that research will bring a solution, maybe in my lifetime, or maybe my student one day, they will find that marker. So that's what drives me to lab every single day.
I never really thought of it from that perspective. I would always say, like, I always assumed that cancer research, the reward would be just like finding the answer, but like I guess the chase of it really is interesting. It makes it more sweet when it. Oh, Joyce, you did say something? Oh, I guess not. What's like the biggest challenge of conducting research today? Sorry.
Biggest challenge, I would say that limiting funding. So limitation of the funding um, is very, very challenging at this time. There are many challenges other than that, but to me, that's a fundamental problem, that if you can come up with 5 different experiments to answer a question or to test your hypothesis, but if you do not have enough funding, especially the kind of research I do, all the consumables we purchase, those are very expensive. uh, and I need someone, meaning my student, my trainees, to work in the lab. They are intelligent, they are bright, so they should be working more. Like my professor asked me to be in research. I find very, very smart and intelligent student in my laboratory. I want them to spend more time in the lab space, but they have other responsibility. They have to maybe earn for their family. So if I cannot pay them stipend or scholarship, that research is not attractive. Meaning that you need to pay training, you need to purchase all those biologics. At the same time, you need to have all the instrument or at least money to buy those services. So to me, uh, cutting down more and more budget or limiting budget towards research, if you do not encourage, you do not have an answer. Okay, we invested 20 years, 30 years behind this vaccination. So now it seems like that 1 year having a COVID vaccine is amazing, sometimes unbelievable. No, you are not looking at those 30 years of research people have done to understand the fundamentals of the vaccination, right? So we have a solution right now. So if you do not invest in research, you are not encouraging basic research. If you are not encouraging basic research, discovery cannot happen, right? So I think, to me, limitation. And also uh, point B would be that to drive curious mind towards research, we have to make research look interesting. As I said, from where I did my master's and PhD, I got scholarship, right? That was one of the positive point, that I can pay my bill instead of working in, in other franchise. I can have my interest answer and earning as well. So if that support is not there, we need to make research attractive. And that's possible, I think, if we give more accolades, more money, more scholarship towards the student who are accepted to the program.
Mm-hmm. And like research, it's absolutely necessary. I don't. It's interesting how there isn't a lot of funding to it when it's so crucial to, like, if we didn't have research and funding, we wouldn't have had this vaccine so quickly for
Yeah.
COVID.
So you think that 1 year is nothing? There was a lot of controversy that there was no step compromised. All you have the fruit of 30 years of research
Mhm.
done already to know about this virus. This coronavirus strain could be new, but the virus itself is not new. So we knew about the virus already. We knew about viral diseases. We knew about how to make vaccine. What about those? Those knowledge cumulative came up, and now government, entire world invested so much of money on that development of vaccine. See, the result came up within a year.
That's really
So if you
interesting.
yeah, so if you invest more, I think discovery will be encouraged more. If I can pay like really $2,000 per month to student, why not? They will be working in my lab. I'm sure about that, right? But to pay them that, I need that grant money as well.
I know bad could come of having, like, there's nothing wrong. There's no negative side effects of having extra research being done because knowledge is power.
Absolutely.
Yeah. The next question is, what would you say are the most significant challenges that students may meet in your course? And what course do you teach? Sorry.
I teach cancer cell biology. That's my specialty. And also I teach genotoxicology. So these are the two upper year courses I teach other than the master's course, which is a seminar-based course on environmental science. So for my specialty, I think that in these courses, you know, one thing is common in these three, two, fourth year and the master's course, that students need to write assignment with their own idea. For that, you need to study hard. You need to search literature. You need to do some literature research, right? To come up with an idea which is not already known or not already being tested or not already being published. They find it very challenging because seems like that once I tell them how to do it, they find out maybe from one topic, breast cancer biomarker, there are 2,000 articles to read. They find it very challenging. So come up with an idea by educating yourself or by training yourself, having that knowledge and come up with something of your own is actually, I'm trying nothing but stimulating their curiosity and trying to push their, research mind within them. I'm trying to push that so that they go through the process, they know how it works. Developing an hypothesis takes months, sometimes year. And that development of an hypothesis means that you need to know everything in that field to come up with an idea, discuss with your supervisor. So they are not left alone. They come to me with 5 different ideas, exciting. They want to address all. Then of course, I tell them, go this way, go that way. But they need to come to me first. So in all these three courses, that's for sure common that they need to write assignment and also presentation. What kind of presentation? Scientific article presentation. I think you will agree, Lauren, that you can present your own work, maybe very confidently, but you will find it very challenging, like to present my work.
Absolutely, yeah.
So these are the two things I find that the student they feel challenged, but they feel so happy and proud once end of the term we achieve those outcomes. When they actually write, that they can see that they're the one who is writing, not me. They're the one developing hypothesis, not me. I'm just channeling their ideas. And when they, with confidence, they're presenting in front of the class, I think that's similar to your speaking public, right? Some students have challenges, so I take care of that in a separate way about public speaking, or they have other challenges. So other than that, in general, students find it very rewarding at the end of the term when they actually finish those. And uh, and I saw many dropping just because there is a presentation of scientific article.
Really?
uh, Yes, because that holds a lot of mark. And as I said, sounds easy, but once you think that, oh, okay, I just not presenting the paper, I need to participate in question and answer session And if I do not know that paper upside down, then it would be challenging for me to answer that. So if you see that writing, presentation skill improvement, and also in genotoxicology, I have one-on-one interview, which might be nothing to do with directly from my lecture. I'm asking you that, can you tell me why we have a significant increase in breast cancer in past 5 years? What do you think? What is your opinion?
Like,
And they, yeah, I'm not asking you. I'm just
okay.
giving you an example that in one interview, they need to face me for 15 minutes, where it's overall opinion build up with the knowledge they gain, that not necessarily you need to digest those lecture to answer me. So development of an idea to write an assignment, public speaking, where you are going to be scored by everyone, and that will affect your GPA. They find it very challenging. And then one-on-one interview, I would say that I had student actually having shiver before coming to even Zoom. Then I deal with them in a separate way. But in real life, you need to face interviewer wherever you go. Doesn't matter. Wherever you go, you need to face an interviewer, right? So I try to help developing those skills alongside with their knowledge in cancer and also about environmental effect on diseases, etc.
Wow, that's really when like the knowledge you learned, you apply it. That's really where it comes together, because that's where you find out if you really learn anything or not, and if you thought for yourself. Yeah.
And those skills helpful every way, every field you go.
Mm-hmm. So did you choose to do stem cell like cancer research and breast cancer? Does it have any close ties? Like, is it personal, or you just took an interest in it?
Very long story. I'm a storyteller, so I do not know how much time you have.
Um,
Uh,
20 minutes.
okay,
15.
so as I told you, like my interest towards cancer research started very early time during my master's uh, in the Indian Statistical Institute. So there I did my master's thesis on cervical cancer. Then I was accepted to that other incident I told you about, that here you go, your curiosity. That was in head and neck cancer or oral cancer laboratory, where they are asking that, is it necessary that everyone smoking cigarette will have oral cancer? If answer is yes, why? If answer is no, why? That was my PhD, to find out that genetic cause and environment together to find an answer why. Okay. And but the training during my PhD at Human Genetics Unit at Indian Statistical Institute is very, very, very fundamental of whatever I am today. Because they told us and being trained us that if you are going to be a cancer researcher, you go with, if you are moving or if you are selecting or you are applying or you are accepted, always pick, if you're interested in patient related research, then go with the disease which is more prevalent in that area. Then you will have more samples, human samples, tumor samples accessible to you than going for the rare one. Maybe you will spend 5 years and hardly get 10, 20 samples if you're working with some rare organ cancer. So this is why when I decided to apply after my PhD, I decided to apply for postdoctoral position. I was applying to the institute, of course, who has that open position, and happens to be the where place I was accepted in Canada, University of Western Ontario. So that's my postdoc mentor is Professor Lala, Piyush Lala. He is an eminent breast cancer researcher. So he asked me the same question as you are. Why breast cancer you are choosing? I said, because I can see 1 in 10 in Canada. That's the incidence. I'm talking about 2009 when I came to Canada. And I'm saying that this is why, because I am very much driven towards patient-related work. and I want to have more accessibility towards the sample. And here I bring in my expertise that not necessarily tobacco smoking, but also genetic mutation, both is responsible to have a disease like oral cancer. I want to investigate the same in breast cancer. If there is a reason why Canada have significantly high breast cancer patients. So so this is the same answer I'm giving you. That was the reason why breast cancer. And to answer about stem cell, that I would say that I do not want to say that accident, but something I thought I did it completely wrong and become and again, one day something happened, and let me tell you the story. So the stem cell are different kind. I'm not talking about the embryonic stem cell or, the stem cell we know, which is being used for leukemia patient treatment. I'm talking about breast cancer stem cell. So these are stem cell within the tumor. These are very tiny, small portion within the tumor who has all kinds of, or gain all kinds of mechanism to resist from treatment. So patient treated with chemo and radiation, they are completely cured. There is no sign of tumor, but after 5 years or 10 years, they come back with tumor in other organ or metastasis in other. And one of those culprit, which is responsible for that disease revoke, is those small population within the tumor who escape all those treatment. So these cancer stem cell, they are called stem cell because they have unlimited self-renewal capacity. So they can keep dividing, but they can trade all kinds of treatment as well. And very tiny population within the tumor, okay? So you don't have all tumors, everything is stem cell. Biggest challenge, I will come to that later, but let me tell you how stem cell happen. I was working in Dr. Lala's laboratory to find a new therapeutic target, okay, which can effectively treat or reduce or shrink the tumor, breast tumor, without having too much of side effect. That was my project for postdoc. So I was working, the meeting, okay, so are we over 40 minutes now?
Oh, it just said that they removed the 40-minute time limit. Sweet.
Oh, that's nice.
Right on. So that gives us an extra 20 minutes before I'll have to go for my exam.
Yeah, you have an exam.
Sorry about that.
No, that's fine. That's okay.
Sweet.
So I thought my cells, which I'm trying to find that new therapeutic target, I transform those cells, I make them more aggressive so that I can use that drug I find out very effective. Every time I'm doing that, looks like that there is an infection in the cell. Okay, so under the microscope, cells look, how do I say that? It's like look like that there is piling up happening. Usually they are flat, but there is piling up, meaning that within the plastic there is a tumor-like structure. And I thought, I infected my cell, so I trashed that. I did this second time, I find the same thing. Then I remember in another lab, I use their microscope sometime. They work with embryonic stem cell. So embryonic stem cell, they have a particular colony-like structure, okay? That seems like that they're piling up. Those are embryonic stem cell, meaning that they are different from the embryo. And we use those for different other purposes. I'm used to see their picture. And I'm thinking I might have contaminated something from their lab to my tumor cell, which is now behaving like those embryonic cells. They are piling up. Okay, so now I was so upset on a Christmas break time that professor from their lab, she's telling that you look very down. I said, yeah, I'm infecting myself. Seems like because I used your microscope, somehow I mix up two cells. I do not know it, but trust me, I never did that. I'm very careful. She said, what are you talking about? Show me your cell. So before I trashed that, I showed her, and she's a stem cell biologist. Her name is Dr. Lynn Postovit. She is now UFA, University of Alberta. She's very eminent like stem cell biologist. She said that, Mousami, you influence a property in this tumor cell. What kind of property? They are now having cancer stem cell. I'm like, what? She said, yeah, you induce that. What transformation you did? I said, I made them more aggressive because I have the drug to test. So the marker you put that in to make them aggressive is stimulating cancer stem cell. Oh, then she said that do all the tests for cancer stem cell. And the picture background you see, that become the cover story of the most respected journal in our field, Stem Cell. They make that the cover story. And that one of my, those spheroid, which I thought I contaminated myself. So you are looking at cancer stem cell colony here, and different color indicate different marker. So do you understand the importance of that? Those small population of tumor cell, problem is to identify them. If you can identify them, then you can cure it, right? Completely. They are the one remaining at the back and coming, helping tumor to come back. So identification is a big problem for cancer stem cell. So now that entire field changed for me. Now I'm trying the role of that gene, which is called COX-2, and its importance in stem cell regulation. And I find out that, oh, it is actually stimulating cancer stem cell. Now we have at least 8 different drugs which can effectively reduce COX-2. Now my work now, not just therapeutic target to reduce tumor, but also to kill cancer stem cell. So now that paper, this work is actually 6 years of work, showing them that I can induce cancer stem cell and I can have the tumor, and in mouse treated with this drug have no side effect. They are healthy and happy. And fortunately, that drug is actually now on patient, that's being approved by FDA for arthritis treatment, the drug I'm talking about. So that's a small molecule antagonist. So one day in my lifetime, I will find out that FDA approved that drug for breast cancer patient treatment as well.
That's
Yeah,
awesome. That's really
yeah.
interesting.
You have exams, so I don't want to tell you more and more story and make it long.
This makes me want to like read more articles on this because I'm not familiar with stem cell research like much at all, and so hearing you talk about it really opens a lot of doors. Because
I know.
interesting. So, what is your latest research? Is this is like one of the first works you did, or what's the
Uh,
most recent thing you've
yeah,
done?
so the stem cell work I have done during my postdoctoral study, and then I didn't stop it yet. We are still working at Brandon University on that. We are looking at right now, as I told you at the beginning, that our focus is that to find a biomarker. What is biomarker? Something you can, so a molecule you can detect in the urine or in the blood or other biofluids of a patient, and you can predict disease. I can give an example. Blood sugar level. Why do we measure that? What
Blood,
does that indicate? Yeah, blood sugar. What is an indicative of?
an insulin or glucose or sorry, glucagon like rise or low.
Yeah, yeah, we call it diabetes, right? So something like that for breast cancer. So our current research is focused on microRNA. Remember that molecule which I transform the cell become induces more cancer stem cell? So that's not it. When I did more research on those cells, I find out that it is changing entire transcriptome, meaning that what those tumor cell now will be expressing at the RNA level as well, not just cancer stem cell. And because my PhD is in genetics, I had that interest all the time to look at the gene part, look at that, remember gene and environment both together. So now I'm focusing more towards that. What happens if you alter in a tumor, if you have really highly aggressive microRNA expressing in the tumor. So these are small RNA, you have it, I have it. So these microRNA, what do? They regulate gene expression. And how? An example I can give you, like our eye color, things which is embryonically already decided, you do not need that gene to express anymore. So these microRNA switch them off, okay? And that's your endogenous regulator, switch on and off. And tumor again hack that. They bring in those embryonic microRNA high because these ones now helping tumor to grow, like the stem cell. Embryonic cell will divide and grow. A baby is growing and growing and growing. So cancer stem cell, they are growing and growing and growing. So now find out that there are more tools to help tumor to become aggressive, and those are small RNA, which are called microRNA. So we find out there are two microRNA very significantly high in breast cancer, very aggressive tumor. And good part about that, this microRNA on its own can make a normal cell very abnormal, very tumorigenic. And also we can find this microRNA in patient blood. So that fantastic, meaning what? Let's suppose that I find out that in patient blood. So what does that mean? That means that is there a difference between the patient microRNA level to the normal person microRNA level? Those two microRNA, okay? 526B and 655. And how does that will help? Well, if I can find a baseline like blood sugar level, that this load means that no chance, but if this microRNA high expression is happening, you need to go for further screening. So we are now testing those two microRNAs role in breast cancer, how they change their nature if you change that environment, if you give them more stress, if you starve them, if you give them high sugar, what happens to those microRNA expression in breast cancer, alongside how many samples we have to actually test the hypothesis I'm talking about. You can detect breast cancer at very early stage in the blood using these two microRNA. So we are proposing these two microRNA as blood biomarker for breast cancer. And that's what we are investigating right now at Brandon University. And this work we are doing in collaboration with multi-institute. So University of Western Ontario is part of it. London Regional Cancer Program at Ontario, they are part of it. So the oncologist, Dr. Braxton, she's an oncologist where patient consent and everything being done, ethical approval being happened. So she's collecting blood from the patient who are coming for screening, breast screening. So we are testing, and it's an ongoing project. So samples are coming, we are extracting, and we are screening at what margin we can say that, this is the best line. Anything up, that's an indicative of that you are carrying tumor. And right, for breast cancer screening, Canada, we have limitation of 50 years. So you have to be 50 years old to have a screening for that.
Really?
Yeah, you need to be 50 years old to get the screening. And mammogram is a painful technique. So they literally squish your breast between two metallic plates. And depending on the tissue thickness, sometimes we miss to get those tumor. And number of patients having breast cancer under age 50 is rising. So of course, undetectable. So when we are detecting, that's already very late stage. Even though we have a treatment for early detection can save 99% patient, 99. So if we can detect that early, in the long run, you can understand we have all those treatment, not easy treatment, but we have at least the option and time to take care of those patients. So early detection using this microRNA, that's the goal. And we hope in our lifetime, we have a blood testing tool, hopefully.
That's awesome. I had no idea. Huh. Wow. That must be really rewarding. Like if in your career, when you guys find that, that'd be crazy.
It is. And sometimes when any article comes up, I get email from patient. That's very rewarding for me. So they ask me where to go, if they want to participate in those trial. So I give them the information. That's very rewarding for me that, when you have a patient is interested on what you are doing, because, at the end of the day, you have an answer to give to them.
Wow. Unfortunately, I think we have time for one more question, maybe two.
Yeah.
So the last one I think I want to ask you is what career, either, I'll let you choose, either what careers are available for a student majoring in this field, or if you could give us advice on pursuing your field of study, what advice would you give us? I'll let you choose which one you want to answer.
Well, I can answer both. In both cases, I would say that for my area of research, you need to be in biology. So you need to be a biology major or biomedical major, uh, because you need to know about normal thing first before you know the abnormal. So I work on breast cancer, which is an abnormal cellular function. So you need to know the biological part of it. So biology, and also having chemistry minor would be amazing. So I will encourage students to go for bio major degree or biomedical major degree, and they should do more and more graduate studies. Unfortunately, that's not very lucrative. Students are more prone towards professional school. That's okay, medicine, dentistry, optometry, but we need researcher, we need scientists as well, who will be discovering those drug and medicine which you have on your table. Tylenol, Advil, those took 20 years of research, right? So be those researcher, have your curiosity, uh, have that passion. If you're passionate, if you are passionate about something, I do not think you are going to struggle for the success. And if you follow that passion, money will follow you, because that's what happened to me. Okay, so I strongly encourage to students who are curious and thinking of grad school, start early. In my laboratory, I take students even from first year. They're encouraged to participate in research. So talk to the professor, talk to the researcher you are motivated or inspired, or you like the work, or the teacher, or ask them that I'm very interested in, patent development. I want to be a patent lawyer. Do I need to know this? Answer would be yeah. If you are going for a patent lawyer, you need to have a science degree. Then you take a law degree, right? So I will encourage students to be more engaged with professor in their undergrad time. There is no right time when to start. If you are curious, if you're interested, you can talk to the professor as early as first week of your university. You talk to the professor, have an appointment, and trust me, we love to talk about our research because nobody seems like wants to know.
This is really interesting to learn about. I knew nothing before this interview. It's really insightful.
No, this
I
is, I'm
find
like, oh, okay, so they're taking it, they need it for their graduation, and I'm the subject, and how I'm the subject, like what's going on? So I really enjoyed this whole thing. So you tell me now, is there anything else? Oh, one thing I forgot to mention, and I think that's fundamental, and if that message goes to someone other than the student itself, that my parent and my husband, okay, he was my partner before and then husband. So if they did not push me and ask me to follow my passion, I could not reach here. We spent significant amount of time at the university, our mentor, but we have a family who needs to be extremely supportive of what we want to do. Okay, so being a university good student, scholarship student, not going for medicine was extremely challenging decision for me and my family. But I'm glad that they stood beside me, especially my mom, who had only high school degree. She always pushed me to go for a PhD. And my father is that's limit. You need to have doctorate after your name, and that's where you stop. Okay, and same for my partner now, my husband. We did our grad school together at Indian Statistical Institute, extremely supportive and pushing me, right? It's not always uprising. No one sees you when you are down. If you're shining, then only you are a star, right? So that time, everyone needs to motivate and actually fuel your passion. And trust me, if you're very passionate and you like what you do, you don't need to think about your success. That will come for sure. And you do need a mentor, okay? find a mentor who knows about your work or can guide you properly. I think these are the fundamental. Follow your passion, have a mentor, and family and partner support the passion, and vice versa. Because I got that support, now I do the same thing. Whenever someone asks me to give them some advice, mentorship, then I'm open to that. Okay, I do that. I love to do that because I know at some point it is really difficult to motivate that don't go for dentistry, but go for a grad school where you do not know. Okay, first PhD, then postdoc, how many years I need to become a prof and how many jobs are there. So if student finish with, I can quickly finish it up. If student finish with this bio major degree, they're open to academia, right? At the same time, most demanding job in past year, you tell me, who?
Mm,
Healthcare worker.
yeah,
They
healthcare.
all need science degree. What else? Those researcher doing screening of COVID. Who
Mm,
else you need, right? Data analysis. These all came from bio major degree. So industry is open for you, academic is open for you, and of course, all those institute, Cancer Care Manitoba, BC Cancer Agency, they are always hiring PhD holding scientists in various positions. Okay. So I would say that I will strongly encourage students to go for further education and grad school, especially.
absolutely. Thank you so much for your time. This is really interesting because
And
I'm
thank
a
you
pre-nursing
too.
student, so this is
Joyce, I see all your chat here. Thank you very much. Thank you so much. And Lauren, before you guys reach me, I never knew about that. Now I know there are something. I knew some news about BU Cares. I saw some podcast because that being circulated, right? And then I was curious, what is the program they're talking about? I knew Joyce, so I immediately said yes to it. But good luck to you too. I hope this is helpful for you. Joyce, thank you. And keep up the good work. I would say that this and do more of these interview. You are doing good for someone else as well if you're putting that somewhere in public places.
thank you. I'm going to stop the recording now. I hope I don't lose it.